We are discussing the fibroblast growth factor receptor 2 protein. Now, this protein contains 821 amino acids, which are protein building blocks in one single chain. And these amino acid sequences spontaneously fold into two different structures. One of them is these little red spirals over here. They're called alpha helices. And the other are these long yellow arrows that come together to create these beta barrels. Now, we have 21 spirals in this amino acid and 6 beta barrels. Overall, this protein contains a 3D V formation, which is on the surface of osteoblasts. It contains an extracellular component to receive its ligand, FGF, a transmembrane component to anchor it to the cell membrane, and an intracellular component so it can propagate the signal within osteoblasts, increasing expression of bone-forming genes. Now, typically, this protein can only become activated after being initiated by fibroblast growth factor, which interacts directly with Ig-like domain 3, which is stabilized by a disulfide bond between two amino acids as seen in magenta. Now, these are cysteine amino acids, and the exact position of this bond in the protein is position 342 and 278. Now, this amino acid position at 342 is a hotspot for mutations due to its high conservation throughout evolution. And in Krausen syndrome, the FGF2 gene that codes for this protein contains a point substitution mutation in the DNA sequence, which when transcribed to mRNA shows a change commonly in the second base pair of the codon at 342 from a G to an A. This results in the cysteine at 342 to change from a cysteine to a tyrosine. So this disulfide bond over here is no longer occurring. Instead, this unbridged 278 gives rise to a destabilized Ig-like domain 3 and results in ligand-independent dimerization. So this receptor is constantly active, which is overexpressing bone-forming genes.